to define a relevant representation for the data. For how do you represent the gene, the bacterial genome, etc. Okay, the first part is going to be about my work on advancing microbial GWAS, and I'm going to define that right now. If you haven't heard of GWAS, it's a problem to try to identify correlation between genetic variants, whatever that means, and the traits. For example, you can think of finding genetic variants that are associated with resistance in bacteria. And the general procedure for that is something that we use a lot in a lot of different settings, that you have this kind of tables where you have a panel with different strains, for example, these are markers, and you put your variants as rows, and you put one, for example, strain 1 has variant 1, 0, strain 10 has variant 1, it doesn't have variant 1, and then at the same time you have a phenotype of traits you're interested in. And then once you have this, you just use whatever testing procedure you want to find rows, variants that are correlated, associated with the phenotype here. And you see that here, the whole question is, what's a variant in this case? And there are, all the procedure I described is relying on this definition of variant. And for a long time, I think it was not really a question, because mostly people were doing that for human genomes, healthy human genomes. And in this case, there is a standard and a very good way of representing gene variation, which is the SNPs, the single nucleotide polymorphism. So if you've heard of that, between, if you take one cell, a healthy cell, and I try to compare the genomes, they're going to be essentially identical up to a few positions. And so if we use this position, variable position, it's going to capture most of the relevant variation between our genomes. So if this is what we're interested in, healthy human tissues, and this is a good thing to do. But it's not true anymore. For example, as soon as you look at human tumors instead of healthy tissues, because then you're going to have pieces of genome that are going to move around, and it's not enough to just look at SNPs. And in our case, it's also important to note that it's not a good representation anymore if you're interested in genetic variation in microbes. Because microbes have, lots of microfamilies have something what's called accessory genome, which is genes that are not present in every individual in the species. So it's not enough. You can't just align the sequences and look at point variations. You have to say, well, do I have this gene or not? If I have this gene, does it have this SNP or not? It's much more complicated. So you can account for this by saying, okay, my variants are not SNPs, they are presence or absence of genes, for example. People have been doing that with a lot of success. But then what about non-coding regions? Or is there a SNP in the gene? And you can always extend that by adding things to your lists, but you need a list in the first place. And so to work around this, people have been using what's called k-mers, which are short sequences as variants, which is a bit less intuitive, but more expressive in some sense. So instead of saying, does my strain to have this SNP, I'm asking, does it have genome, does it have these big words of size 40, for example, in the APCG alphabet. And if you think about it, by doing that, it's something very expressive because you can capture SNPs and you can capture the presence of genes without having to define any of them. If there is a SNP somewhere in the genome, it's going to trigger the presence of particular words. Same thing for genes. Okay? So you don't have to define them, and you can capture all of them, but at the same time, the results are much less interpretable. Right? I'm going to show why. So here is an example. Let's say that there is a gene somewhere in the genome that are accessory genes that contains a SNP which gives resistance to the bacteria. Okay. If I look at k-mers, I break the whole genome into k-mers. Around these SNPs, I'm going to have just for this SNP 8 k-mers of size 4. Okay, that correspond to the same information having going to contain the same information. If I look at larger words, to improve things, we've been using the brain graphs. Without going into detail, the brain graphs are defined over k-mers, the nodes are the k-mers, and they are connecting overlapping k-mers. It's widely used in assembly and variant coding methods. It's not a new tool at all. It's used a lot in bioinformatics. And for us, it has two main interests. One is compaction. Here, again, without going into detail, if you have this kind of structure where you just have one SNP, it's going to trigger the presence of this kind of structure here, a bubble in the graph. The k-mers that are involved in the SNP are going to be all connected in this form of bubble. And once you have this structure, you can compact it and you can say, well, okay, all these k-mers that contain the C, for example, they actually correspond to the same information. If I have one of them, I have all of them. I can test the presence of this unique larger word. So I eliminate some local neighboring structure because indirectly this structure is going to represent the genomic context. That is, if I look at the k-mers that are nearby this one, we're still doing the test on the k-mers, but try to make more sense of the results. And it's work that has been mostly done by Magali Jayar and Leandro Rishi during their PhD. Okay, so the small cartoon I was showing you before was a very small graph on a few k-mers. So it was very simple. And you could say, well, I can just look at the graph and see what's happening. More typically, if you try to build the brain graph on a full set of genomes, there are genomes, you obtain this kind of picture, so you can't look at it manually. But what we do is remember that each node of the graph is a k-mer, a short sequence, okay? Or maybe a longer sequence. And we can do one test for each of these sequences. So we're going to do one test for each of them, and we're going to retain only those that are significant. That is, the words whose presence in the genome is associated with the phenotype of interest. And we're going to look at the neighborhoods in the graph of these significant nodes, and you see that sometimes some of these neighborhoods will touch. And we are going to extract the connected components that are generated by this process. Is we're going to group together the k-mers that are hopefully nearby in the genome. So this is the kind of component that we obtain with the tools. Remember that the nodes are essentially k-mers. It can be a bit longer if I have some compression. The edges are overlaps. And here I'm using colors to show the percentage of resistance traits that contain this word. So here, if I have red nodes, it means that most of these k-mers are present. These k-mers are present mostly in resistance traits, more than sensitive traits, for example. And so what we see is that we capture this kind of almost linear graph. If it was linear, it would have been compacted during the compaction process. And it's representing a polymorphic sequence, the kind of thing I was talking about. It's not linear because there are small variations that trigger a lot of small bubbles like this. And if we look at what these k-mers correspond to, it's the top hit for the subgraph here is top hit for Staphylococcus aureus medicine resistance, and it corresponds to a well-known resistant gene. And this is the cassette part in which the gene is inserted. So if you look at that for the first time, it may look very cryptic, then you don't see how it helps in any sense. But just remember that the alternative before this tool was just to look at a very big list of unstructured k-mers. We just have a huge list of K-mers, you don't know how to make sense of them, and this is saying, well, at least all those, you should look at them together, they probably correspond to a single polymorphic sequence. So if we circle back to what I was saying, I said we should use the brain graph to structure the K-mers. So have we done that? We've done that in the sense that we've provided a higher level interpretation. Once we have selected significant K-mers, we can say, oh, we have subgraphs that can be interpreted as genes or plasmids, for example. But we're still making the test at the K-mer level, at the node level of the graph, which is not very satisfactory because we're doing the test at one level and then we're doing the interpretation at a different level. And also, maybe more concretely, you may have a power leak because, like I said before, you're testing. If this is one gene and you have different versions of them, you're testing separately the different versions, so you're losing power. So the next part I'm going to present here was work done by Hector Roux-de-Bézieux to try to use the graph to define new variants. Okay, not test variants and then interpret them using the graph, but use the graph to define variants. So here is a very small toy example where you assume that you have a causal like a gene that's giving resistance. That's accessory. Not everybody has it, but it exists in 4 versions. It's a 4-letter gene. It's very simplified. And all of them have GA in the middle, and then you can start with A or T and end with A or T. If you draw the brain graph of the 3-mers of the gene, then you obtain something like this. And then what if you try to do the previous procedure? Okay, so here I have 4 strains. Each of them contain one version of the gene, so, and they're all resistant, and then the ones that don't contain any of these versions are sensitive. If I test the K-mers separately, you see that each of them will account for only half of the resistance strains. For example, TGA is only present in TGAA and TGAT, but not in the 2 others. So the profile is 1, 0, 1, 0, whereas I want 1, 1, 1, 1. I want perfect correlation. If I manage to do something that's presence of this gene in any version, and I should have 1, 1, 1, 1, and more power for the detection. So in the best case, if I can do that, and probably it would work. In the best case, if I have enough power, enough samples, for example, then I will detect that these K-mers are correlated with resistance, and then DBGWAS may say, oh, look, they all connected together, so you should, that's a single unit. But like I said, we have more power to detect something that would be A or T, then GA, then A or T again. And if you look, it's just a logical OR over all the K-mers in this subgraph, the brain. So maybe it's even harder to interpret in terms of genetic variants than a K-mer. In this case, having a subgraph variant means having one version of this long sequence, if you want to try to make sense of it. So that was for one very small short example. If we want to do that systematically, again, the graph is huge. We have to test all the connected subgraphs of the brain graph, which is in the millions of nodes, at least. The number of subgraphs is exponential in the number of nodes. So we have 2 obvious problems at first. One is that we will have a critical multiple testing problem. So we're going to if we manage to do the tests, we're going to have a huge number. We will have no power at all to detect anything. And even without thinking about the tests, just enumerating all the subgraphs is going to take too long. I won't give the technical details, but the intuition between what we did is behind what we did is that some of these subgraphs can be discarded without even being tested. It's a concept called testability that was introduced in a paper by Tyrone in 1990, I think. And the intuitive idea is that if you want to test the association with a variant which is present in all of your samples, then you don't even have to look at the phenotype or to do any test. You know that it cannot be associated with anything because everybody has it. It's not technically a variant, okay? Everybody has it. And the idea of testability is to quantify that and be a bit more subtle. Say, well, even if not everybody has it, if too many people have it, even if I don't look at the correlation with the phenotype, it's impossible that it's associated with anything that has significance. So I can discard it. And if you think of it in terms of the, so that would be for one k-mer, for example, and then if I try I start growing a subgraph around this k-mer, you see that the presence profile of the subgraph, if I do an OR, logical OR on all the k-mers, is going to grow. So if the presence profile was 1, 0, 1, 0, and I add anything, I can only add once in this. So at some point, if my profile is not testable, it cannot be associated with anything, then I can stop, because adding will only make things worse. Ouais, je vois les petits, je ne sais pas comment. Clique pas sur le rouge, clique sur le vert. Je peux bouger peut-être. C'est pour un bout du titre. Tu peux pas le déplacer. Non, non, c'est une Okay. So with this idea of testability, we use it to solve both of these problems. The critical multiple testing problem will be addressed because the non-testable subgraphs somehow don't count towards correction. It may sound like cheating, but it's a thorough procedure. It's well defined, so you don't need to count it towards multiple testing correction. And then for the enumeration, you can use the pruning strategy using it. At some point, you can stop enumerating and you manage to enumerate what needs to be tested. And that's one example of what we get with this procedure. The first hit that we get for amikacin resistance and Pseudomonas aeruginosa. And it's a plasmid with resistance genes. If we use DBGOS at the same level, and I think even at much more liberal levels, we'll only recover small pieces of the plasmids that are then hard to reconnect. And it's slower than DBGOS, than testing the k-mers. But the main contribution was a new exploration procedure that used the pruning that I was talking about. And it runs in 3 hours for the whole GWAS, whereas if you use the previously existing exploration methods, you explore only 2% of the same number of subgraphs in 24 hours. So it's the main contribution of this work. Okay, so to conclude on the GWAS part, we've been using the brain graph to help interpret the k-mer-based GWAS and also define a richer class of invariants, including polymorphic genes. The perspectives of that are try to scale up, make things faster and work on a larger genome, more larger genomes, and also further improving interpretability, because maybe if you just look at this graph, it's not that obvious what it means yet. I'm going to move now to another part of my recent research on predicting from biological sequences. So you can think, instead of trying to find the pieces of the genomes that are associated with the resistance, now I just want to predict whether this genome, bacteria having this genome is going to be resistant or not. It's not the application I used, but just to relate that to the previous parts. The work I'm going to present, big part was related to what's called convolutional neural networks, which have been very popular, in particular for computer vision, but in machine learning in general. I will just recall the idea. Well, if you have a sequence, you will start, so here it will be A, T, G, C, G, C. You will start by doing what's called the one-hot encoding of this sequence. That is, each of the letters is going to be associated with a vector, with a 1. Here, A, it's a 1 at the A position, and zeros everywhere else. Okay, it's very standard for sequences. It gives you, instead of a character string, it gives you a numerical matrix, so you can do standard things with that. In particular, convolution and convolution operation is going to use what's called convolution filters here, which have the same number of columns and a given number of rows. Here are just 3. Okay, and you're going to slide each of these filters around this and take dot product. Okay, it's the convolution operation. Intuitively, each of these dot products is going to tell you how similar this part of the sequence is to this convolution filter. In the end, you're going to obtain for each filter convolution profile, the similarity of the sequence along the sequence to this filter. We explain how you can interpret these filters after. Then you typically apply a nonlinearity. Here I just put the negative parts of 0. Pooling along the sequence here I just take the max. So that's the highest similarity along the sequence to the convolution filter. And then this gives you a new representation of the sequence that you can use with maybe a linear model to give you a prediction. So these are the two parts that you learn in the network. You learn the convolution filters, you optimize them for best prediction, and you learn the weights on this representation. So in some sense, you're learning at the same time the representation, a good representation of the data, and then a classifier acting on this representation. So like I said, it's been originally used for computer vision, and it's known that at least for the first layer, you can interpret the filters, the convolution filters, as small image patches because they act on pixels. So they are homogeneous to the pixel, and you can interpret them as a small image of the same size. So and what you obtain in general when you train your model is this kind of shapes where you are saying, well, what's important is to capture the presence of an edge, for example, in a given direction. If you do the same reasoning for the sequence, the one-hot encoded sequence, you can get some kind of interpretation as a probabilistic sequence motif. You obtain a small matrix, and instead of having a 1 for A and then zeros everywhere else, you just have real numbers. You can, for example, say they need to be positive and sum to 1. In this case, here at this position, you will have mostly a T, but then it can be A sometimes as well. Okay? You're just relaxing the one-hot encoding because it's just a real set of real numbers. So in this sense, I said that the CNN, the network, was learning the convolution filters. If you believe in that, then learning the convolution filters is equivalent to selecting a set of probabilistic sequence motifs that provide a good representation for prediction, optimal for your prediction problem. And so if you think in these terms, it's not that different from k-mers, right? It's just a way of, it's a continuous relaxation of the k-mer. The k-mers could be encoded, so it's a one-hot encoding with ones and zeros. Here it's just a relaxation of that. So before that, when we had polymorphisms, we used the brain graph to represent these polymorphisms. It was a small connected subgraph. It's a discrete way of representing that. Here, we're going to do the same thing, but with a continuous representation. So I like to connect these two in this sense because I think it's, for me it was interesting to see this connection. But just to be honest, it's not exactly the same thing because in particular, CNNs are using relatively short filters in general and acting on relatively short sequences, whereas I was using k-mers of size 40 on full genomes for the other one. So conceptually, you can think of these are two different versions of the same idea, but in practice, they are not used in the same way at all. And the first contribution I'm going to talk about was done by Dexiong Chen during his PhD. It was a kernel method point of view of the CNN. So it was one of the contributions was to connect these networks with what's called positive definite kernels. See more. And so try to understand this idea. We start by building a representation, a different representation. Okay, it's also giving you the link with the k-mers I was talking about before. This was the representation by k-mers. Okay, for this is the one sequence, and if I have AATC, I put 1 here. Then this is AATG, for example, etc. 1 or 0. You could think of a relaxation of that. Instead of using the k-mers, I'm using all possible probabilistic motifs. So I have infinitely many here. I can store that or represent that in any way. But if I have AATC in my genome, I will have a high value at the AATC level of the function, and then maybe smaller values on similar motifs. So you cannot represent it, but you can compute the inner product in this representation. There is a way, without representing the full infinite vector, to compute the inner product implicitly between two such vectors. And that's the idea between won't say more about positive definite kernels, but kernel methods are about that. They are often about saying, well, I can build very rich representations. I never have to actually store it. All I need to do is being able to compute these inner products, and then lots of learning methods only need, they can work only with these inner products. So I'm fine in this case. So you can do that with this representation, but it's still slow. And also, it may be a good representation, but it will not select important motifs. It will use all of them. And to change that, we started from something called Nystrom approximation. So Nystrom is a classical tool on kernel methods that used to approximate the inner product. And the idea is that you rely on a representation of each sample by its similarity to a small number of other samples. You could say I just pick a few k-mers, and then instead of representing my sequence by this infinite vector, I'm just going to represent it by its similarity to AATG, TCGG, etc., or a few values. So it gives me a small finite vector, and I need to compute to be able to compute this inner product for these few anchors. But once I've done that, I have a new representation, and somehow magically, if you take the dot product in this new representation, it's a good approximation of the original dot product. Okay. So it's a well-known tool in kernel methods, and it solves the problem of we could use the previous representation much faster by just doing that with scalars, for example. But it doesn't select motifs, for example. It doesn't select features. And so what we use instead of just sampling randomly k-mers as anchors, as the points to which we computed the similarities, we also optimize the anchors for prediction. And if you do that and you write it down, it formally leads to a form of convolutional neural network. Okay, so you recover the convolutional neural network by doing these two steps. First, learning an approximation of your representation by optimizing anchor points, and then building a classifier over its representation. That's the same structure as before, right? And the optimized anchors, of course, are your convolution filters. So for me, just that is interesting because it helps me understand what CNNs are doing. But in addition, we don't get just any CNN by doing that. We have somehow a more regularized version. So if you know anything about machine learning, you know that very often, especially when you don't have a lot of samples, you want to enforce some kind of regularity on your function that you're learning. Say, I don't want this function to be arbitrarily complex like this, because otherwise there is no way I'm going to generalize on new data. And without going into details, this method is a way of regularizing the convolutional neural network, inducing regularization. Okay, and so, like I said, it's showing that training a CNN is a way of formally a way of selecting probabilistic motifs. So if this is selecting probabilistic motifs, maybe the natural question, given what I said before, is can we do hypothesis testing, where we've selected features, right? So can we do more than just selection and put a test on that? That's what Antoine Villiers did during his PhD contribution. So again, if you take a very simple one-layer convolutional network, it's formally selecting the motifs that are maximizing some score. Detail that, but it's quite straightforward to write for some associated some score between the motif and the phenotype. But then how do we know that these motifs make any sense? It's like if you think in terms of GWAS, you could just look at the correlations between my k-mers or my SNPs and the phenotype, and then even if I don't have any signal, if I take a randomly simulated phenotype, I will always get some correlation. So that's why people do tests. That's why people don't stop there, and they need to have to quantify the uncertainty. But we don't do that when we select features with CNN. So we'd like to do that. Say, okay, I have selected motifs. I want to test the association. For example, say my phenotype is some true parameter plus some noise. Is this theoretical true parameter associated or not with the motif that I want to test? And if you've heard of that, you can feel that there's an issue here because we've already used the data for the selection. Right? We've used the data to train the CNN. We've done selection, but in this sense, we've already used the data. And if we use the same data again for testing in any way, not in any way, but in a naive way at least, the p-value is not going, the test is not going to be valid. It's going to be over-optimistic because you're quantifying the association between your phenotype and something that you specifically selected for its association. And there are different ways of doing that. You can just split your data. It's the easiest way, but then you lose some power because you just use part of your data. And then there's a family of methods called selective inference, where you instead of using the null distribution, you use the null distribution condition on the selection event. But then all the methods that exist only work from finite sets. And here, the training the CNN is making a selection from an infinite set of motifs, if you want to think of it in these terms. So we did something very naive. We just meshed the space of motifs. So it's a huge space in this case, but it becomes finite. Instead of saying I can have any point here between A, T, C, and G, some 3 dimensions, and just repeating meshes. So I have a finite set of possible values. And then the association between the phenotype and the mesh that has been selected. We also have contributions on the actual test itself that I won't tell it here. And so the procedure that we have, if we do that, is more powerful than doing data split. So it's better if you have few samples, because you don't need to use half of it for selection and half of it for testing. But it's much less scalable. It takes much longer between you need to somehow do a Monte Carlo Markov chain to estimate your functional non-distribution. So this makes the two methods complementary. If you have a lot of data, you can use data split. You won't lose that much power. If you have less data, then you can afford the more powerful method. So contributions on this part and neural networks for sequences have been to cast links between neural networks and kernel methods, and also make very simplified at this point CNNs amenable to statistical inference. Again, the perspective would be to do the same thing on whole genomes, for example, for the prediction and generalize the testing procedure to more complex networks. I'll move to the third part of my recent work, which is more recent and so more preliminary, which has been on using machine learning for simulation-based inference in molecular evolution. So that's the evolution part I was talking about at the beginning. And all this work has been done by Luc Dassel, who is a postdoc working with me now, Vincent Garot, who just finished his master's internship, Lucas Nesterenko, who is a PhD student, and Johanna Trust, who was an engineer. Okay, inference in evolutionary genomics. I don't know how familiar you are with the problem, but the general setting for that is that you observe homologous sequences, meaning sequences that evolved from a common ancestor. And from that, you want to infer their evolutionary history in a broad sense. So it could be, I will focus on the parts that I look at in my work. One is the phylogeny. So you want to build the actual tree over which those evolved. You don't observe this tree, just observe the tips. Or parameters of the evolutionary history, and I use that for epidemiology. So it's R0 that you may have heard of in early infectious period, etc., etc. And the issue is that very often in evolutionary genomics, the models, you have probabilistic models, very, very good probabilistic models, but they are complex, and it's hard to even compute the likelihood sometimes, let alone optimize the likelihood. So it's often either impossible to compute the likelihoods or sometimes very expensive. And you have a trade-off. If you want a more complex and therefore more realistic model, very often it's more expensive. So because of that, people have been using for some time something called simulation-based inference, which allows you to do inference without even computing likelihoods. The most well-known and maybe older version of that is called ABC, Approximate Division Difference. The idea in the context of machine learning is that you're going to simulate, maybe your model is too complex to compute likelihoods, to optimize likelihoods, but it's simple enough to simulate data. That you can do. If this is the case, then you can simulate lots of examples. In our case, for phylogenetic, for example, we'll simulate trees and then sequences evolving along these trees. We can do that. And then we're going to use this data. We're going to use this for supervised learning. We're going to learn a function that takes as input the sequences that we observe and that outputs the evolutionary parameters that we don't observe, but that we know for the simulation. Thanks. And so, in some sense, this is generalizing what's done in ABC, where you simulate samples with known parameters, and then you retain the parameters that gave samples that are similar enough to your data, and you say, this is my post-real. This is very similar to that. And the advantage of all these approaches is that they are fast, and they can deal with more complex models, as long as you can simulate data. One of our contributions for that is that we've been working on aligned sequences, and so that's the kind of thing I was showing you before, right? They all evolve from the same common ancestor, and so you align them, and you see that some positions are identical for everyone, or almost identical, and then some are variable, and that's what you use in general for inference with these methods or likelihood-based methods. If you want to use machine learning on this, you want to learn a function that takes these kind of alignments as its input. And you can see that it's important to have some kind of invariance or equivariance, depending on the problem, to permutation of the sequences. Because let's say you want to predict a tree for this alignment. You don't want the tree to be arbitrarily different if you just permute the sequences. What's important is the set of sequences. It's not order in which you get sequences. If you want to predict the Arnold, for example, if you have a set of SARS-CoV-2 sequences and you want to predict the Arnold, you don't want the value to change depending on the order in which you put the sequences. And if you've never thought about this problem, this has no reason to be true in general. We use, for example, one hot encoding like before, and each of these sequences, and we stack them, and we use a linear model on that. You can see that if I just permute the first two sequences, I'm going to obtain arbitrarily different results. Because I have a weight for the first feature, a weight for the second one, etc. The model doesn't know that it should be invariant by permutation. And so we're going to rely on a very popular mechanism that's called self-attention, that's been used in transformers, for example, to retain some expressivity. The idea being that just being invariant by permutation is not that difficult. You could say I just average. I have several sequences, I do one hot encoding, and then I average all of them. That's my representation for the alignment. Then I have invariance, but intuitively I've lost some signal. Self-attention will allow to retain more information than that while leaving invariance. Okay, these are some of the results that we obtained. One, the goal was to obtain to reconstruct the tree topology from the alignments. So we were generating trees using what's called the birth-guess process. And then once we have these trees, we use a classical simulator and model to evolve sequences on this tree. We get 200,000 alignments of trees of 20 sequences and sequences of length 200. Sorry, this plot is not very up to date, but the idea of what we obtain, the orange is the best possible method so far, is the likelihood method. That's expensive. And the green one is the result that you see that we are not as good as likelihood, except and well, we are better on the setting in which we are training. It's not obvious on this plot. I didn't choose the right plot. Sorry about that. But the message that we have for the moment is that we managed to be actually as good as, or sometimes better than likelihood, if we train on 20 sequences and test on 20 sequences. but if you try to move away from that, the model is well defined. Attention can be trained on 20 sequences and tested on 50 or 100. But we lose some performances, and I'm not sure why yet, but doesn't think are changing when we change the setting simulations. So for the moment, yeah, for 20, we are as good or better. Around that, it's still defined, but it's less efficient. And the important point is that it's much faster. It's about 100 times faster than the likelihood methods. It's slower than the existing, more naive approaches, but then again more efficient. That's the first result we have for phylogenomics, where we don't reconstruct the tree, but we just predict R0, for example. Here again, we have Burgess sampling tree because we assume that we don't observe all the possible sequences, we just observe them. Then we evolve again on that. We train again alignment of this time set of 50 sequences. In this case, the state-of-the-art method is Bayesian estimation inference method called BITS. And we get about the same performances for predicting the parameter of the physics, so R0 duration, but it's much faster. So I'll be very careful about this result first, but also because here I'm comparing in terms of accuracy. This is not just doing the prediction; it's giving a posterior distribution. So it's not directly comparable, and one of our goals is to see how we perform in terms of reconstructing this full posterior, not just the prediction. So that would be uncertainty quantification, and then simulate under more complex models, because this one is basic. And I think that's it. I just want to thank, I already acknowledged some of these people along the presentation, but most of this work has been done by students and postdocs, and then some of my colleagues with whom I worked. Thank you for your attention.